Hello students, welcome back. So here your biology teacher Raminder Kaur. We are going to start with the topic of chapter number 10, Microbes in Human Welfare. Name of the topic is Microbes, how they are useful in industrial products. In the first video lecture, we have studied that how various household products such as curd, cheese and many other bakery products are made with the help of microorganisms. And today we will study how at the large scale in the industries commercialized products are made with the help of various microorganisms. Two examples are given in front of you. First is beverages. Beverages can also be known as alcoholic drinks. Number 2 is given antibiotics. We all know when there is an infection of bacteria, always antibiotics are given. These two things are prepared with the help of microorganisms. And now the next page, you can see 10.4 figure where you can see a big containers or vessels. These are known as fermenters. If you remember, two kinds of respirations are there, aerobic and anaerobic respiration. In the absence of oxygen, most of the glucose, that is a sugar, converted into alcohol. So, number first is fermented beverages, in which wine, beer, whiskey, brandy, rum, all these kind of alcoholic beverages are made. Now, which microorganism is used? This is the brewer's yeast, used for the production of ethanol by fermenting malted cereals and fruit juices. Mostly, the fruit which is used is grapes and malted cereals. Let's study what is the meaning of malted cereals. Any of the variety of a cereal grain, it can be rice, wheat, oats and rye, can be used to make malt. The most common by far, however, is barley, bajra, which is the primary grain used in the production of most beers and majority of malted whiskies. Here malt means when it gets fermented during anaerobic respiration, it makes a malt. So here the yeast, which we call as a brewer yeast, its scientific name is Saccharomyces cerevisiae, which we have studied in the first video lecture also, used in bread making. Now the question comes, do you recollect the metabolic reactions which result in the production of ethanol by yeast? So we have already talked, it is either ethanolic or alcoholic fermentation which takes place when sugar converts into ethanol, carbon dioxide and other byproducts. Now, depending upon the type of raw material, I'm again telling you raw material can be fruit juices or it can be any malted cereals, and the type of processing. Now, if I talk about how petroleum crude oil is being extracted, we are using diesel, petrol, kerosene. It is how it is done? Yes, fractional distillation. There are two kinds of distillations. One is destructive distillation and second is fractional distillation. This is a kind of technology by which we are extracting various liquids at different boiling points, right? So either with distillation or without distillation, we can prepare different kind of alcoholic drinks. Here it is written, being highlighted, wine and beer are produced without distillation, whereas whiskey, brandy, rum are produced by distillation methods. You can see the picture of fermentation plant. So these are the big containers or vessels. They are also called as fermenter vats, VATS, which are used for distillation process. Now, on the next page, I have shown the process of wine making. So here you can see that the fruit juice from the grapes is first of all crushed, then it is being extracted, it is pressed, horizontal press are there, and then it is again basket presses there, and now different kind of wine is made. So after the destemmer, the wine is pumped into tanks to begin fermentation. So microorganism is used. Which one? Saccharomyces cerevisiae. And this process of fermentation, it turned the grape juice into an alcoholic beverage. During fermentation, yeast transforms sugars present in the juice into ethanol and carbon dioxide. Let's come back to this page. I hope this fermentation beverages topic is clear to you. Now we will come to the next topic, that is antibiotics. So anti means against, bio means life. So something which is against the life. Against the life means which can kill some living organism. Here antibiotics definition is given. I have underlined. Antibiotics are chemical substances which are produced by some microbes. It is not a single microbe which is used. There are different kind of bacteria which are used and can kill or retard. Retard means stop. Cease, C-E-A-S-E, means which can stop the growth of any other disease causing microbe. Here note down, disease causing microbes are also known as pathogens. First made antibiotic is penicillin, but its discovery was by chance. The scientist Alexander Fleming, he was working on Staphylococci bacteria. You remember yesterday in the first video lecture we have talked about that spherical bacteria, they are called coccus. So here the name of the bacteria is Staphylococci. When he was working, he found that a mold was grown on his unwashed culture and that where Staphylococci could not grow. He found out that it was due to the chemical which was produced and he named it penicillin after the mold Penicillium notatum. So this was established by Ernstine and Howard Florey, and now it is extensively used and even it helped people in World War II in America. So let's take out a brief of this antibiotic discoveries. First of all, Alexander Fleming, he discovered it. Penicillin was extracted from Penicillium notatum. Notatum is a species name. It is used to cure many bacterial diseases. Then streptokinase. Second one is streptokinase. It is extracted from Streptococcus bacteria. Where it is used? It is also used in clot buster. You know, whenever there is some injury, if blood continuously flows, then there can be death also. But we have special cells in the body. If we talk about blood cells, we have RBCs, WBCs and platelets. All three are present in a liquid known as plasma. So those platelets are acting as clotting factors. So here also, if our body does not produce specific amount of platelets, then we can provide this streptokinase. So streptokinase is one kind of antibiotic, also an enzyme. That's why it is ending with the name ase. So it can remove the clots from the blood vessels of the patients who have undergone myocardial infarction. Myocardial infarction means those in which heart surgery is being done. So we have discussed now antibiotics, wine making. Now they have asked, can you name some other antibiotics and find out their sources? Yes. So kindly note down. Penicillin we have studied, extracted from Penicillium cryocyginum, notatum also, and this species also. Then second is erythromycin. Usually we intake this Streptomyces erythraeus. Then third is streptomycin, extracted from Streptomyces griseus. Last is tetracycline, extracted from Streptomyces rhinosus. Then they have talked about that how they were useful in history. First disease was plague. Note down, it is caused by the bacteria Yersinia pestis. Scientific name, it was zoonotic bacteria. Students, zoo term is always used for animals. Means the infection was caused by animal. Zoonotic bacteria. Found in small mammals and their fleas. People infected from this bacteria develop symptoms after an incubation period of 1 to 7 days. Like we are talking about now coronavirus, that 14 days quarantine is done if somebody develops symptoms in these days. So incubation period means till the time when symptoms appear. There are two main clinical forms of plague infection, bubonic and pneumonic. Bubonic plague is the most common form and is characterized by painful swollen lymph nodes. We all know that there are blood vessels as well as lymphatic vessels. So nodes are those places, those tissues where all those lymphatic vessels are joined or connected. So they can appear in arms, they can appear in our legs, and it can lead to more infection. Second type, which is not explained here, is pneumonic. Pneumonic, we all know, it can lead to pneumonia. Now next disease is whooping cough, Kali Khansi. It is a highly contagious means which can communicate easily, respiratory tract infection that is easily preventable by vaccine and is particularly dangerous for infants. Then number third is diphtheria, Galkotu. In diphtheria, it is a serious infection of nose and throat that is easily preventable by a vaccine. What happened in this case? A sheet of thick grey matter covers the back of the throat, making breathing very hard. So it can lead to a problem. There is a risk in breathing. Last one is leprosy, Kushi Rog. In this, it is a chronic. Chronic, you all know, there are two kinds of diseases. One are acute and second are chronic. Chronic are those diseases which take place for a long time. They are curable, infectious disease, mainly causing skin lesions and nerve damage. So there is skin infection. Leprosy is caused by the infection with the bacterium Mycobacterium leprae. So scientific name, when you write, must underline them separately. So effect can be on the skin, eyes, nose and peripheral nerves. Peripheral nerves, you remember, there are different kind of nerves, like various nerves are there which are taking the signal to the brain as well as spinal cord, and many are bringing back. So spinal and the peripheral nerves. Next is in industrial products. How can various chemicals, enzymes and other bioactive molecules produced? So you have to note down, you can learn this only with the help of a chart. So here I am with chart. This can help you in learning. Number first, they have talked about few acids. First is citric acid, second is acetic acid, third is ethanol, and last is lactic acid. Number first, citric acid, which microbe or microorganism is used for making it? Aspergillus niger. Underline when you write. It is a category belong to fungus, that is a yeast, used in beverages. Number 2, Acetobacter. We are using very commonly vinegar in our kitchen, right? It can be apple cider vinegar or different kind of vinegars are available. Actually, it is an acetic acid. Category, it is a bacterium Acetobacter, and role, it is used in preservative. Then number 3, ethanol. Saccharomyces cerevisiae. We have talked about so many beverages. It is a kind of a fungus and used as disinfectant or fuel. Number 4, lactic acid. It is extracted from Lactobacillus, kind is bacteria, helpful in making curd. So with this, we have completed this number first paragraph. If I repeat from here, we have done Aspergillus niger. It is making citric acid. Acetobacter aceti, aceti is species name. It is making acetic acid. Then one was not given there, that was butyric acid. So Clostridium botulism, it's a bacteria, butyric acid is made. And last is Lactobacillus, a bacteria that is making lactic acid. Now in the next paragraph, we will talk about various kind of enzymes. The enzymes which are useful in our daily life. How are they being made? Number 1 is lipases. They are used in detergent formulations and are helpful in removing oily stains from the laundry. So whatsoever surf you are using, any detergent, Surf Excel, Tide, all these are containing this enzyme lipase. Number 2, whenever we are bringing any fruit juice, it's very clear. When we are making juice at home, it's not clear, rich of fibers, but when we are bringing from market, it is very clear because it is clarified with the help of pectinases and proteases enzyme. Then number 3 is streptokinase. We have already talked about it in antibiotics case also. So it is extracted from Streptococcus bacteria, which is useful in clot buster for removing clots from the blood vessels of the patients who have undergone myocardial infarction leading to heart attack. So we have till this completed. You can see Streptococcus bacteria that can lead to streptokinase. Then Clostridium we have already covered up. Now we will talk about number 3rd paragraph, in which they have talked about several kind of bioactive molecules. Number 1st is cyclosporin A. Where it is used? It is used as immunosuppressive agent in organ transplant patients. How many different kind of transplantation takes place? There can be various organs which can be transplanted, but how the body detects that it is its own self or non-self cells. So if we are transplanting, let's take an example, kidney is being transplanted in any patient. Body can reject the kidney which is coming from outside. So during that, these immunosuppressive agents are provided, that is cyclosporin A, so that body could not reject it but it can accept it. Here, what is the function of immunosuppressive agent? That it is reducing the immunity so that antibodies should not kill the kidney that is coming out from outside from any donor. So from where this cyclosporin A is extracted? That is extracted from a fungus known as Trichoderma polysporum. I hope this is clear. If I repeat this, cyclosporin A is used as immunosuppressive agent. Immuno means immune system, suppressive means which can stop. So when it is used? When there is a transplantation. From where it is extracted? It is extracted from a fungus, Trichoderma polysporum. Then number 2nd bioactive molecule that is very important is statins. From where it is produced? It is produced from Monascus purpureus. I am repeating how to pronounce. You have to listen very carefully. Monascus purpureus. So it is used as blood cholesterol lowering agent. Mostly we have seen there are several patients who are saying that my cholesterol is high in the blood. So those medicines, they contain these statins. It acts by competitively inhibiting the enzyme. If you remember in biomolecules chapter class 11, we have talked about how enzymes can act as inhibitors. So here this statin is inhibiting the enzyme which is responsible for synthesis of cholesterol. So I hope this chart will help you a lot. So they have also talked about these statin and cyclosporin A. I feel there is spelling mistake in 7th point. So you must note down it as statin. So here enzymes we have talked about. Number 1 lipase used in laundry detergents. Number 2 pectinases and proteases which are used in bottled juices. Number 3 is streptokinase that is extracted from Streptococcus bacteria used as clot buster to remove the clots. Now our next topic is microbes in sewage treatment. Sewage treatment plants are useful to clean water. So various kind of bacteria are useful. Those microorganisms are helping to clear that water so that it can come to the position that it can be disposed of in ponds, in lakes and any other water bodies. So here they have talked about major component of this wastewater is human excreta. So this municipal wastewater is also called as sewage. We have seen So those areas which are coming under municipal corporation, they have these sewage underground. So before disposing of, we need to clean it. So this sewage treatment plant is also called as STP, so that it should be less polluting. Here you can see this chart can help little bit. There are two kind of sewage treatments. Number one, primary treatment, followed by secondary treatment. In lower classes, you have studied that if sand is dissolved in water, how can you extract sand? Yes, it's easy. First of all, you have to keep it for some time. What will happen? Sand will lie at bottom. Then you can filter that water and sand will be remain at the back. So here also, this primary treatment consists of two techniques, filtration and sedimentation. So when sand settle down at the bottom is called as sedimentation, and the upper residue we can filter it, and that filter is sent for secondary treatment. So on the next page we can see primary treatment. So this treatment step basically involve physical removal of particles. Two methods are there, filtration and sedimentation. What do you think? Which kind of waste can be removed by it? All the floating debris. DEBRIS. Debris means all the excreta, human, animal excreta. Everything will settle down. So it is removed by sequential filtration. Sequential filtration means with the sequence, step by step it is removed. After that, soil and small pebbles. Pebbles means stones. Small stones, they are removed by sedimentation. All the solid that settle down, they form the primary sludge. Everything which has settled down on the bottom, that is called as primary sludge. And the supernatant. Supernatant means a liquid that forms a effluent. Now that effluent which is produced from the primary settling tank is sent for secondary treatment. I hope this topic is clear to you, and in the third video lecture I will start with secondary treatment, that is also known as biological treatment. Thank you.